In this video, I'm going to go over the process of human development. I'm going to go through it kind of quickly because you pretty much have all the notes that you need, so you can always look back at your PowerPoint and get the information that you need. Some definitions: pregnancy, here's gestation period. It just gives you the human gestation period. I'm not going to be asking you specifics about that, but a little bit of trivia: the longest gestation period is in a blue whale. I think it's a blue whale. But elephants actually have a 2-year gestation period, which is very long. Some definitions: from the time fertilization till 2 weeks is a zygote. 3rd through 8th week is embryo, and beyond that is a fetus. We've talked about mitosis in depth, going through prophase, metaphase, anaphase, and telophase. The goal of mitosis is to end up with 2 cells that look exactly like the first cell. In meiosis, which only happens in the ovaries of females and the testes of males, you have to end up with 4 cells that have half the chromosomes, so 23. Because remember, what's going to happen is the sperm's going to donate 23, the egg's going to donate 23. So meiosis is kind of, not really, but kind of like 2 mitosis phases, but instead of ending up with exact duplicates of the first cell, you end up with 4 cells that have half the chromosomes. In a male, you do end up with 4 sperm with 23 chromosomes. In a female, like you learned in lab, you only end up with 1 ovum that has 23 chromosomes. So meiosis occurs in the testes and the ovaries. Fertilization. When ovulated, the oocyte has this zona pellucida around it, and it also has this corona radiata, this area here. So this is actually part of the follicle that came with the oocyte when it was ovulated, and that should be a little bit of review from lab. So what has to happen is the sperm's going to have to get through both of those. So millions of sperm are ejaculated in the vagina, but very few, the proud, make it the 5 inches to the oocyte, which for a sperm is like running a marathon. So it's a very long way. Many are killed by the vagina, the acidity in the vagina. They have to get through the mucus to get into the uterus, and so very few of them actually make it.
Failure is not an option.
But the ones that survive are very determined to make it. So here's one of the few thousand sperm that makes it to the oocyte. Even though these sperm have made it, the sperm is still not ready to fertilize the egg yet. They have to get to the actual ovum. They have to be what's called capacitated, which is not decapitated. The membranes in that acrosome have to break down, and then when the acrosome breaks down, it releases these enzymes that are going to break down the outside of the egg. So we've got this. All these sperm get there. Okay, all these sperm get there. They break down their acrosome, and then those enzymes are going to break down that corona radiata until one comes in contact with the zona pellucida, which is like the membrane, and then that sperm is going to be the one that fertilizes it. So this representation is a little dramatic, but this gives you an idea what's going to happen. So one guy gets in, the one that hits that zona pellucida first. That'll be the one that's going to fertilize the oocyte. And then the other guys are mad. These are just a couple cartoons you can look at, and you can look at those on your PowerPoint slide. So fertilization, one gets in.
Tranquility Base here. The Eagle has landed.
So what'll happen is when the sperm gets in, to prevent many sperm from fertilizing the egg, the fertilized egg puts this, think of it as a brick wall around it. It's called a cortical reaction. So no other sperm can get in. This is probably people's first baby picture, and someday I'm sure we'll see one on Facebook. But what happens is until the sperm fertilizes the egg does not finish meiosis. And so this takes a little bit of time. So the sperm gets in, that signals the egg to finish meiosis to get down to 23 chromosomes, and then what you have here is the 2 nuclei from the sperm and the ovum fusing together, and then immediately after they fuse, then mitosis will start. So when the egg is fertilized by a sperm, That signals it to finish meiosis, and then as soon as the nuclei fuse, then it will begin mitosis very, very quickly. It actually gets a special name, and that name is called cleavage, because this process of mitosis happens so quickly. And because of that cortical reaction in that brick wall around the fertilized egg, the cells are going to get smaller and smaller because the developing zygote is not getting any bigger. So cleavage is fast mitosis. The cells are becoming smaller and smaller. At this point, it is heading down the fallopian tubes or the oviducts towards the uterus. This shows it at 2 cells, at 4 cells, at 8 cells. You get to this point, okay, a few hours, 72 hours after fertilization, and you have what's called a morula. So these cells at the morula level, let me go back, are undifferentiated. What that means is they can become the whole organism. So if we separated these 4 cells and put them into 4 different petri dishes, they would continue to go through mitosis. They would continue then to form their own little zygote, and you could implant those back into whatever animal they are from, and you would get 4 different organisms that were genetically identical. So all these cells have the ability to become the whole organism. When cells get into a more mature state, they are differentiated, and that means that they are going to be a specific tissue and a specific organ. So they will have certain songs. They will be a pancreas cell, they will be a stomach cell, they will be a brain cell. They will be more differentiated to do the job they are meant to do. So with in vitro fertilization, what they do is they watch this process of cleavage that occurs in a petri dish after they put a sperm and egg together under a microscope. And then once they get to this phase, this is where they will implant it back into a woman's uterus. If it looks like this, then they will not implant it, because that means it's not going through the process correctly because of how it looks, and that it will not survive. So we have a good one, we have a bad one.
Houston, we have a problem.
So when this occurs again, they will get rid of these. And these really don't have the ability to become a full developing fetus because it's not going through the process correctly. Day 4, we have what's called the blastocyst. So now you start becoming a little differentiated. This area is going to become the placenta. This, which is called the inner cell mass, is going to become the developing fetus and embryo. So what are twins? There's monozygotic twins where you have 1 egg, 1 sperm, and then as it heads down the oviducts or even into the uterus, it splits into 2. And so they are genetically identical. The earlier they split, they may have 2 different placentas because they will implant into different areas of the uterus. If they split later, as opposed to sooner, then they may share a placenta. This can sometimes cause issues. If they're sharing a placenta, one of the developing twins may be getting less blood than the other. Then we have dizygotic twins. It just happens that the woman ovulates 2 eggs, 2 different sperm fertilize them, and you end up with twins who are no more alike than siblings. The Olsen twins, actually, believe it or not, are dizygotic. So you can have a male and a female. With identical twins, since they come from 1 egg and 1 sperm, they have to be the same gender. Conjoined twins are where would be coming from a situation where the egg and sperm it's been fertilized and it starts to split, but it doesn't split completely, and they end up sharing organs or being attached in some way. These twins, you may see them on Discovery Channel. They are conjoined twins. They cannot be split because even while they have their own hearts and nervous system, trying to replace or build back a whole half a body would almost be impossible. At day 6, it implants, so that means that it's going to dig into the lining of the uterus. It's then going to develop a placenta by the end of the 3rd month that will be fully mature. You have the amnion. The amnion is a sac. You hear about the amniotic fluid, or you hear about woman's water breaking. This is this amniotic sac breaking. It's going to do all these different things: protect from trauma, homeostasis, prevent embryo growing parts to go together, As the kidneys mature, the fetal urine will be put into this amniotic fluid. All right, for the next section, all you need to do is you need to know the highlights. And so just learn when these things happen. Notice here we have a tail to start with. We have little buds as flippers kind of things, as arms and legs. You'll notice how big the head is. The nervous system is one of the first things to develop. Here you can see the rib cage starting to form. 8 weeks, it looks like an alien. Cardiovascular system's fully functional, limbs are present. Fingers and toes are webbed, and all the brain parts exist at this point. That's why if a woman takes drugs, using alcohol, smoking, it's going to have the most dramatic effect if she's doing that early in the pregnancy when these systems are starting to develop. 12 weeks. Here's the highlights. At 12 weeks, you should be able to tell the gender. You can tell the gender before this if you do a genetic test, but to actually see the developing reproductive organs, you won't see that till 12 weeks. Skin is formed. 14 weeks. Here's what it looks like. 18 weeks. Lanugo, you get a silk-like hair that covers the skin. They get the fetal position because they're getting too big to be sprawled out in the uterus, so they'll start to come into the fetal position. The limbs are going to start being a regular proportion to their body, and this is when the woman will start to be able to feel the fetus moving. At 6 or 7 months, they look like little old men. Period of most significant weight gain. The eyes will begin to open. Myelination, so nervous system is going to start to mature as that myelin starts to form. And for males, the testes are going to descend into the scrotum. Remember, testes and ovaries are formed from the same tissue up in the abdominal area. In females, they will descend slightly, but if they are testes, they will descend to the scrotum outside the body. 8th or 9th month, last system to fully develop, we've done this, is the respiratory system. They are pretty squished there in the uterus. Braxton Hicks contractions late in the pregnancy. These are kind of practice contractions done by the mother's uterus. A lot of times with the first pregnancy, the woman will think she's in labor, go to the hospital, and they will usually send her home. Parturition is the technical word for birth. This is a Body Worlds exhibit of a woman who died 8 months into her pregnancy. She knew this was a chance that this could happen. We think she had diabetes. They never told us what, but there's always a chance with the diabetic that the mother and child will not make it. This fetus is incredibly big, which is also another sign that she may have had diabetes. So she had donated her body prior to, or right at the beginning of her pregnancy, knowing that something could happen because of her medical situation. Stage 1 is dilation. Oxytocin, prostaglandins initiate labor. Maybe signals from the lungs that start this process. Stage 1 lasts from the time of labor until the cervix is about 10 centimeters large. This can vary in time. First round through with the first child is usually the longest. Expulsion. I'm not sure if this is the right.
Put me back in, please, buddy. Let go of my head and put me back inside. Ah!
So this is from full dilation to delivery of the infant. Crowning, largest part of the baby's head. Episiotomy. They don't do these as much anymore, but in some cases they will actually cut from the vagina down towards the anus, not all the way through, but down, to allow the baby's head to pass through more easily. They have found that this actually causes more pain for the female afterwards, and it actually heals better if the tearing would happen naturally. Stage 3, delivery of the placenta. Usually happens 30 minutes after delivery. This has to be all removed. One of the major dangers of childbirth for females is bleeding after the birth. And part of that can be because they don't get the placenta out. So that has to be removed. It will be removed naturally, but the doctor will usually go in and make sure that all of that has been removed from the uterus. And then you have this last stage. This is basically, takes a long time. Everything's kind of going back to what it was before the birth process, so the uterus is going to shrink in size. It will be larger than it started, or be a woman who has given birth will have a larger uterus than those that haven't, but again in this stage it will start to shrink and go back to as small as it's going to get after that birth. So that is human development and the process of birth. You just need to know highlights. You need to know what I've gone over, and that will be on the final exam.